It is important for you to keep these lab handouts for your lab quiz that's coming up about mid-semester. These will be the primary things you will use to study for that lab quiz. And it is not uncommon for us to ask some of the same kind of questions that were asked in the things you were supposed to turn in. So all I did in grading them was kind of give you a sheet, and if you missed any points, I indicated what points have been taken off, but I didn't write in answers on all of those. So I want to do that now. So please correct any questions you got wrong, so you have that information. On the chromatogram, we have in this particular chromatogram 4 known amino acids. And it's hard to see, but the little spot is in the center that allows you to determine RF, which is the distance traveled by the spot divided by the distance traveled by the solvent front. What some of you didn't do, just a few people, but some of you, is this was supposed to be a mixture of 2 or more amino acids. So in that little chart there, you should have had RF values for at least 2 unknown spots. Some of you had an RF value for 1 unknown spot, and then said there were 2 or 3 different amino acids in there. That can't be the case. In this one, there are 1, 2, 3 spots here, so there should have been RFs indicated for each one of those, and then you compare those to the knowns to see which one you have. Okay. So, some of the questions, let's run through real quickly right here. Regular soft drinks sweetened with corn syrup with a lot of glucose. Diet drinks with NutraSweet, which is a dipeptide. So which 2 tests? Read the question carefully. Some of you only gave me 1 test. If you did the Benedict's test and it was positive, that would indicate a regular drink because glucose is a reducing sugar. The diet drink should give a negative result because it doesn't have glucose. Then you should also do the Biuret test because the dipeptide will give you a positive Biuret test. The regular drink should not give you a positive dipeptide test. Okay, so just didn't form it very well. So I said, what if a student proposed having a test for hydroxyl groups for reducing sugar rather than doing the Benedict's test? Waters are negative control, and assuming that there's an available test for hydroxide groups, is this a good idea? Well, it's not a good idea for a number of reasons. Many of you said this would not differentiate between reducing sugars and non-reducing sugars because carbohydrates have hydroxyl groups, and that's entirely true. But another problem is that water has hydroxyl groups. Remember, water dissociates into protons and hydroxyls. And if that were indeed the case, then water would not be a negative control. You would get a positive hydroxyl test for your negative control. So be aware of that. Medical evidence about whether chicken soup would be good for someone who has a cold. That basically just asks you to go back to your data. Did your results indicate a positive or a negative Biuret test for your chicken soup? Okay, fats are made up of glycerol and fatty acids. Both of these contain polar groups. And so glycerol has 3 polar groups, and then each fatty acid has a polar group, which is the carboxyl group. Okay? So both constituents of a fat have polar groups. So why are fats themselves not polar? Because we get this reaction that occurs between the carboxyl and the hydroxyl. And when that reaction occurs, water molecules are liberated, and a covalent bond then forms between the glycerol and fatty acids. Okay? Anybody know what this bond's called? It's called an ester bond. Okay, so the polar groups become involved in the ester bond. Student RF of 1.2, not possible. That would indicate that the amino acid moved further than the solvent front, and the solvent's what is transporting the amino acid. More than likely what students did was they divided the wrong way. Okay, and this is a result we've gotten in the past sometimes, and so we like to throw that up there as a scenario that actually has happened. And then finally, student says they messed up because when they're all done, their chromatogram is completely blank. Okay, so what's the problem here? Well, for some reason, they either didn't get any amino acids or enough amino acids on the paper to detect. That's one possibility. Or maybe they actually ran it all the way off of the paper Although that wouldn't happen if the solvent front were before the end of the paper, but that's a possibility. Maybe they submerged the spotted amino acids below the solvent and it washed away before the chromatography really had a chance to get going. Even if amino acids were on there and separated by the solvent, if you don't dip it in anhydrin, you can't see the amino acids, so it would look blank. Okay? Or if you didn't heat the paper, this reaction of anhydrin with amino acids requires heat to occur. And so those are some of the possibilities for what could have happened. So please make those notations and then save these so that you have something to study for your lab quiz. And I will give you a study guide for your lab quiz as the lab quiz approaches, so you've got an idea of kind of what's going on here. Okay, this week we're going to do several things in lab. We're actually going to do 3 experiments simultaneously. The first one that you want to get started is the effect of temperature stress on bead membranes. In this particular experiment, you're going to take beets, and beet tissue is loaded, I said in class the other day, with this pigment anthocyanin. And we're going to use a cork bore to bore out little chunks of round beet tissue, and then cut those up into little pieces and expose those to different temperatures. The idea being is that different temperatures might cause stress on those beet membranes. And the more stress that occurs and the more damage that occurs to the membranes, the more those anthocyanins will leak out of the beet tissue into the surrounding fluid over time. How are we going to measure that? We're going to measure how much fluid leaked out, how much pigment leaked out, using the spectrophotometer. And you guys did the spec lab last semester, correct? Okay, so you should all know something a little bit about the spectrophotometer. Okay, a couple of things to be careful about when you do these experiments. When you take your cork bore, push it into the beet, and then there'll be a little stick of some kind that you can stick inside the cork bore to push the little segments out. And the segments are longer than you'll need. So you can use a razor blade and cut them into the appropriate length. I can't remember how many centimeters they are, but it specifies how many centimeters long those should be. You can put them in a little petri dish then, get a little bit of tap water, and then just swish those beet segments around in the tap water to wash any pigment off that was released when the tissue was prepared. So when you push that cork bore through that beet, you're going to tear some of that tissue. So we want to wash off the surface pigment that's leaked out from those damaged cells. We've got several test tubes, and we're going to put them at different temperatures. The most extreme temperatures, the 2 cool temperatures, the freezer or the refrigerator, we actually take the beet segments and put them in an empty test tube and let them sit at that temperature for 30 minutes. The other mid-range temperatures, we're going to take the beet segments and we're going to expose them to a little bit of water of various temperatures in a little beaker. But they're only going to be in that water for a minute as opposed to 30 minutes. Why the difference in time? I don't only need a minute exposure to the mid temperatures or even the high temperatures, but the low temperatures require longer exposure. Any ideas? We're trying to heat the tissue, right, and expose the cells to different temperatures. Is there any difference in terms of heat transfer between liquid and the tissue and air in the tissue? It's going to take longer to cool it down if it's just sitting in air. There's less heat transfer between the air in the tissue than there is water in the tissue. Okay? After you expose those to their various temperatures for either a minute or 30 minutes, whichever is appropriate for your particular situation, then you're going to take those little pieces out and you're going to put them in another test tube with 5 mils of water and let it sit for 30 minutes. What's going to potentially happen during that 30 minutes? If there's damage by that temperature, then the pigments are going to start to leak out into the solution. And after 30 minutes, that will be enough time for any pigment to leak out into the solution. After 30 minutes, we pour the solution off, and then we will read the absorbance in the spectrophotometer. Once you have poured the solution off after this 30 minutes, you can read the absorbance of that solution anytime. Okay? Now that the tissue's out of there, it stopped leaking. So this can sit until you're ready, and it's a convenient time for you to take those readings. When each group has taken their readings, what you're going to do is there will be a chart up on the board that has columns for -5, freezer, +5, refrigerator, and then these 4 water bath temperatures. And then each group will record their absorbance data for each of those particular conditions. So this is group 1, group 2, group 3, etc. Okay? You are then going to have to, prior to next week, plug this data into StatBook, because we want to know what the mean and standard deviation is for each of these temperatures. So this is all the class data for each temperature. Because ultimately we want to see is, are there significant differences between the means? Did one temperature damage the membranes more than the other? So if you put that data in the StatBook, it'll look something like this. Okay? So here's the data you entered, and it will calculate for you the mean and standard deviation. This is the mean, this is the standard deviation. Now, the question you're going to ask is, are any of these differences significant compared to the control? The control will be this 25 degree Celsius temperature right here. Okay? So this has a mean of 0.24, an absorbance of 0.24. This had a lot more pigment leak out into the spectrophotometer tube. That's why it's got higher absorbance. It's redder. Okay? But is that difference significant? Can you make that determination looking at just that data? No. So you'll also then have to look at the t-values. And these t-values will then allow you to compare each of the different temperatures to one another to see if there are significant differences. Remember that when you make that comparison, this calculated t-value has to exceed the one in the t-table, which is in your lab book, for at least the 95% confidence level for the appropriate degrees of freedom. So if I want to compare this one to this one, what degrees of freedom do I look at in that t-table? What's the appropriate degrees of freedom for comparing -5 to +5? Pardon me? Remember the definition of how you calculate degrees of freedom. It is 12. Remember, n-1 plus n-1. So n-1 plus n-1, 6 plus 6 is 12. So you would look at that 12 degrees of freedom row, the 95% confidence level, see what that expected or tabular t-value is, and then compare that t-value from your t-table down here that you calculated and see if this is greater. Okay? So we're going to get that started first. Second part is a simple experiment on the permeability properties of a model cell using dialysis tubing. These are the materials that you can use. We had asked you to develop a written procedure that you would bring to the beginning of class period. Unfortunately, we had not changed the date from last semester, so it said Monday the 24th instead of Monday the 23rd, so you caught a break. Okay? But what we're going to ask you to do in class today, before you take the quiz, is quickly huddle with your lab partner and try to come up with a procedure. When you're done, you can come up and grab one of these sheets, which is a quick kind of representation of a simple procedure to use, and then compare yours to this one. Then you can take the quiz and then you'll be done. So work on your procedure with your partner quickly. Come up and grab one of these when you're done, and then once you've talked that over, you can take the quiz. Now, a couple things to point out. This is what you have to work with. You only get one piece of dialysis tubing per group, so please keep that in mind. It's some thread, a couple of solutions, A and B, and then you've got stuff to run the Benedict's test, the Biuret test, a balance, And then you get some test tube, Sharpie, ruler, boiling water bath, plastic cup, Pasteur pipette. What we're trying to determine is, what is the permeability properties of that membrane? Is it permeable to water? Is it permeable to iodine, protein, glucose, and starch? And you can set up a really simple experiment with one piece of dialysis tubing that will allow you to determine that. So we'll have you work it out in a second. Okay, last thing we're going to do is movement of substances into and out of yeast cells. Here are some things that you'll be using in the experiment. We will use a dye called neutral red. And neutral red is a pH indicator. It has a different color depending upon the pH that it's in. At an acidic pH, it's red. At a basic pH, it's orange-yellow. We're going to be working with sodium carbonate, which is a base. We're going to be working with an acetic acid, which is obviously an acid. During this particular experiment, you're going to take various solutions of yeast and sodium carbonate and neutral red and mix them together, do some filtering, and determine what these particular yeast cells are permeable to and how they're moving those substances into the cells. When we filter, you're going to have a fairly large volume, like 25 milliliters. It's not necessary to filter the entire 25 milliliters to get the results you need. You need to collect a little bit of liquid, that is the filtrate, and so you can look at its color. And then trapped on the filter paper will be the yeast. And as long as you've got some yeast cells trapped on the filter paper, then you can tell what color they are. So don't waste time trying to filter all the solutions. When you acidify solutions, you can just take a little bit of the solution you want to acidify, and you can just eyeball it. We're not even going to measure it with any kind of pipette or anything. And just add an equal volume of acetic acid to the volume you have already. And that will accomplish acidifying. Whoops. Why we've set this up so that the procedure is shown as a flow diagram. You're going to be working with two flasks, A and B. This shows you the steps and the procedure that you do to each of those flasks. So for example here, mix yeast and sodium carbonate, boil it on a hot plate, then add neutral red, split the sample in half, filter some of that, and then at various key points it asks you to record color. What color are the cells? What color is the solution? And there's little numbers right here. Those little numbers correspond to your data sheet. Okay, so for flask A it says initial color of the suspension, 1. Well, that is this step right here, 1. Okay, what color is that initial suspension? Over here in flask B, you're going to do similar things. One of the big differences is this is boiled and this is not boiled. So these are going to be dead cells. These are going to be living cells, basically. Okay? Well, you're going to collect the data and put it into this data sheet right here. And in some cases, the results are not applicable. You don't need that observation. But once you've got this information, you can answer some questions about yeast cells. Okay? Like, is the membrane differentially permeable? Is there dye inside the cell? Did sodium carbonate enter the cells? You can deduce this by looking at your results. This is a great exercise in deductive reasoning, and also a great example of how you can learn a fair amount about the properties of the membrane of this yeast cell by mixing it with a few chemicals and filtering. It's a very simple but elegant experiment. And then kind of the big question that really tests whether you understand this or not is answering this question based upon this data. Is the cytoplasm of a living yeast cell acidic or basic? Now, you're probably going to need some help on this one, okay? There'll be parts of it and you'll go, I don't know what I'm doing. At that particular point in time, Dr. Morgan will try to guide you through it without giving you the answers directly, okay? But he'll help you reason through that. I can tell you right now, it's really important for you to be able to understand what you're doing here. Because on the lab quiz, there will be a similar type of question. It'll give you a similar but different type of experiment. with slightly different results. Okay? So the answers to these questions on the lab quiz might be different than they are because you get different results presented to you. And then you're going to have to draw conclusions. So don't just have your lab partner fill this out and memorize the answers and then think you're good to go for the lab quiz. You need to understand the logic behind this. Okay? Anything I forgot?
Nope.
So start the beats first. Once the beats are started, then you can get your dialysis tubing set up and get that going because that has to sit for 30 minutes. And then once that's going, you can get started on this. So you need to be organized, okay? So it's a good thing to have read through this ahead of time. So if you would get with your lab partner and turn to your lab exercise and see if you can come up with a quick procedure for the dialysis tubing experiment. When you're done, come up and pick up our little diagram and then make sure you leave enough time to take your quiz. Okay?
Should we have them pick up the quiz when
Yeah. You got yours already.
Yeah.
I'll just give you this one then, and you can share this with your partner so you can compare that.
Partner's not.
Okay. Who's your partner?
John.
Okay.
Thank you.
So you can just take the quiz anytime.
Can I ask a quick question?
Sure.
Go ahead.
Oh, I need to do I need to bring my procedure?
Did you have it already done?
Yeah.
Okay.
Ligands, is that just a general term for all signaling? Because in the book, it kind of, in some charts, it labels ligands and some it didn't.
so whatever you have, biological terms, protein, something else.
So a substrate would be a ligand for an enzyme. So it's a molecule that specifically binds to another. Want your quiz?
Oh, yeah.
Thank you.
Sure.
Big challenge is to learn this.
Yeah.
And then to realize that nuclear tells you things about age.
Yeah.